Assalamu alaikum. Monosaccharides. Monosaccharides are the simplest carbohydrates that cannot be further hydrolyzed. They are soluble in water and sweet in taste. They are composed of polyhydroxyaldehydes, so they are known as aldoses, or polyhydroxyketones, so called ketoses. Monosaccharides can be classified either on the basis of carbon atoms or on the basis of their functional group they contain. So on the base of carbon atom, they are trioses having 3 carbon atoms, just like glyceraldehyde and dihydroxyacetone. Tetroses having 4 carbon atoms, just like erythrose. Pentoses having 5 carbon atoms, e.g., for example, ribose and xylose. Hexoses having 6 carbon atoms, and they include glucose, galactose, and fructose. Hexoses, we are mostly familiar with hexoses. So heptoses having 7 carbon atoms, like pseudoheptylose. Now coming towards the classification of monosaccharides on the basis of their functional group. So they may be aldoses having aldehyde functional group, or they may be ketoses, ketonic functional group. Aldoses example is glucose, and the example of ketoses is fructose. Now remember that the most abundant monosaccharide in nature is the glucose. Oligosaccharides, the second class of carbohydrates. So they are composed of 2 to 10 monosaccharide units, and they are linked by a covalent bond known as glycosidic bonds. So as they have many units, so that's why on hydrolysis yield corresponding number of monosaccharides types. They may be disaccharides, which means that they have 2 units. They are composed of 2 units. Disaccharides are most common in our diet, and they include sucrose, which is formed by the glycosidic bond of glucose with fructose; lactose, which is formed by the glycosidic bond of glucose with galactose; and maltose, which is formed by the glycosidic bond of glucose with glucose. Now trisaccharides having 3 units are 3 monosaccharide units, and so on. So usually trisaccharides and other tetrasaccharides and pentasaccharides, they are found in glycoproteins, just like blood grouping antigens, etc. So one point to be noted that oligosaccharides attached to proteins or lipids are essential for cell recognition. Why they are essential for cell recognition? Because they act as a cell surface markers, they act as a receptors. So that's why the hormones or other signaling molecules usually attach to those receptors, and also they help in identification or recognition of cell. That's why it has a role in our immunity as well. Coming towards polysaccharides, also known as glycans, they have high molecular weight. They are complex carbohydrates and made up of more than 10 monosaccharide units. Typically, they are insoluble in water. They are non-sweet and non-reducing types. So they can be broadly classified like homopolysaccharides and heteropolysaccharides. So homopolysaccharides, also known as homoglycans, they are called homopolysaccharides because they are made up of same type of monomers or same types of monosaccharide units. Example: starch, glycogen, cellulose, aniline, and dextrine. Glyco okay. Glycogen is highly branched, allow rapid glucose mobilization. Now coming towards heteropolysaccharides, so they are also known as heteroglycans. As the name indicates, it contains different type of monosaccharide units or derivatives. Important examples are glycosaminoglycans like heparin, chondroitin sulfate, hyaluronic acid, dermatan sulfate, keratan sulfate, or blood group polysaccharides. One clinical point to be noted that if there is a defect in degradation of glycosaminoglycans, it can result into conditions known as Hurler syndrome.